Okay, before break, we talked about glycolysis as the first part of cellular respiration. And this is an overview image showing the reactions that are a part of glycolysis. And just to review, you start with 6 carbon glucose, it's split. It is phosphorylated and then split into 2 3 carbon sugars. You have the um, using of 2 ATP or consuming, and you have the production of 4 ATP. So there's net 2 ATP. And there's also a reduction of NAD plus that occurs to produce NADH. The next part of this that we are going to be looking at, and of course the slide will not switch, and I don't know why. The next part of this is that we are going to talk about the Krebs cycle. And in the Krebs cycle, um, well, before we go into the Krebs cycle, we have to take care of pyruvate. If you recall, before break, we also talked about the fate of pyruvate. And in anaerobic respiration, if there is a lack of oxygen, pyruvate from glycolysis will then go into fermentation. And that all is without oxygen. But if oxygen is present, then the pyruvate will be oxidized and it will be brought into the um, uh, Krebs cycle. So in glycolysis, it's glucose to pyruvate. But since there are still bonds, carbon to carbon bonds in pyruvate, there's still more energy that can be released. And so um, there are 3 more carbons that have to be stripped off of pyruvate. In other words, to oxidize. So if oxygen is available, pyruvate will enter the mitochondria. And in there, the enzymes of the Krebs cycle complete the full oxidation of sugar to CO2. Can I call you right back? Bye. That was my husband. Um, and then we have pyruvate as it is breaking apart the carbons. What will happen is that those carbons will actually leave into the environment as CO2 and released. So there are carbons in pyruvate that still need to be removed or broken apart, and that will then release the carbon dioxide. So there's 3 carbon pyruvate is broken down into 1 carbon carbon dioxide. And there you can see how glucose is broken down. But there are a lot of things that occur in conjunction with all of this, and that's what we're going to talk about. So, um, overview again, you have glycolysis, and this is in the cytoplasm. You have the mitochondria that consists of the Krebs cycle, the electron transport chain, and oxidative phosphorylation. You have ATP produced in all 3 parts. Notice here that the ATP is produced by substrate level phosphorylation, as it is also here. If you recall, we talked about what substrate level phosphorylation is. Simply that phosphates are added from substrates, intermediates, molecules, to ADP to produce ATP. Um, and then we'll talk about oxidative phosphorylation a little bit later, but that is another way to make ATP. And then if you look across the top here, you have NADH is carrying electrons to the electron transport chain. NADH and FADH2 will carry electrons from the Krebs cycle. So the first thing you have to remember is that it is important for you to understand the structure of the mitochondria. And it is a double membrane, and it has a smooth outer membrane, and then the inner membrane is highly folded. And those folds are called cristae or cristae. And then in between, there is the intermembrane space. And we colored this, so you should remember it. It is the fluid filled space between the membranes. The matrix is the inner hollow area here in this image. And it is a fluid filled space. Mitochondria have their own DNA and their own ribosomes. And they have enzymes that are free in the matrix as well as those that are membrane bound.